Up till now, we have discussed that the photosynthesis process involves two reactions, that is the light reaction and dark reaction. Photosynthesis involves the light reaction and dark reaction. Right? Now, as we have already discussed, during this light reaction there will be formation of ATP and NADPH taking place. Apart from that, there will be a process taking place called as photophosphorylation. The dark phase is also called as a biosynthetic pathway, where there will be fixation of carbon dioxide taking place. So now what we will discuss is, we will discuss the process of photophosphorylation in detail. How the photopigments and photosystems that we have studied in the last class will be involved in synthesis of these ATP and NADPH. Now, let us first understand the term photophosphorylation. As you can see here, there are two terms. One is photo and another one is phosphorylation. As I said earlier also, phosphorylation will be termed because there will be fixation of a phosphate group to form ATP molecule. And since it is taking place in the presence of light, the complete process is termed as photophosphorylation. We have also studied that in photosystem there are two types of reaction centers, or there are two types of photosystems. Photosystem 2 and photosystem 1. Photosystem 1 absorbs 700 nanometer of light and photosystem 2 absorbs 680 nanometer of light. Now, this process of photophosphorylation can actually take place in two different ways. So, there are two types of photophosphorylation reaction. One is the non-cyclic photophosphorylation and the other is cyclic photophosphorylation. Non-cyclic photophosphorylation and the second is cyclic photophosphorylation. The non-cyclic photophosphorylation involves both photosystem 2 and photosystem 1, whereas cyclic photophosphorylation involves only photosystem 1. So we'll one by one study in detail the non-cyclic photophosphorylation and then the cyclic photophosphorylation. So the first is non-cyclic photophosphorylation. Now, as I said, these processes will be taking place at the reaction centers. Now, the non-cyclic photophosphorylation starts with actually a reaction center having chlorophyll A molecule which absorbs light of 680 nanometer. So you can say that the non-cyclic photophosphorylation starts with PS2. Surrounding these reaction center, there will be of course the other accessory pigments which are going to absorb the lights of different wavelength and pass it on to the reaction center chlorophyll A molecule. But the chlorophyll A molecule here is itself involved in absorbing the 680 nanometer wavelength of light. Now, as soon as these absorption is being done, there will be a pair of electron that is released. Since the light is absorbed, the electrons will get excited and these excited electrons will be transferred to a primary electron acceptor. which is associated with photosystem 2. These primary electron acceptor will always be associated with the photosystem 2. After the pair of electrons is being accepted by the primary electron acceptor, they will be transferred to a electron transport system. So the 2 electrons will now be transported to the electron transport system. The first complex, there are different complexes which are involved in the electron transport system. The first complex here is plastoquinone. PQ is going to represent plastoquinone. The plastoquinone is going to transfer these pair of electrons to the next complex of the ETS, that is the cytochrome complex. CYT stands for cytochrome complex. There will be different types of cytochromes which will be involved, and that is the reason it is written here as cytochrome complex. Now, the electron from these cytochrome complex will now passed on to the last complex of the ETS, which is PC. PC stands for plastocyanine. Now, when these pair of electrons is being transferred from cytochrome complex to the plastocyanine, what happens here is one molecule. The energy which is released by the transfer of these electrons will be utilized by this ADP molecule to form ATP. So, we had studied during the process of light reaction, ATP synthesis takes place. So, this is how here the process of photosynthesis is, synthesis of ATP is taking place. Now, the electrons which are transferred from the primary electron acceptor are always transferred in a downhill manner. So, from one electron transport, one complex of the ETS to the other electron complex, it will be transferred. Now, these plastocyanine, now it is going to actually transfer these pair of electrons to the next photosystem, that is PS1. The chlorophyll A molecule of which is going to absorb light of 700 nanometer, and hence it is also called as P700. Here this ATP molecule is being formed. So one of the activities of the light reaction also has been completed. The light energy, here the light of 700 nanometer will be absorbed by chlorophyll A. So this is the photon light which is being absorbed, and then the electrons will be transferred. Like here also these electrons are transferred. What is happening? The light energy which was trapped by chlorophyll A molecule is converted into the chemical energy which is in the form of ATP. Now these two pair of electrons, as I said, which are transferred by the plastocyanine, is going to be absorbed by the PS1 photosystem, which is also going to absorb the light from atmosphere, right? So that light will be of 700 nanometer, and these electrons which are transferred here to the reaction center of the second photosystem, or you can say PS1 photosystem, will now get excited and transfer that pair of electron to its primary electron acceptor. So the two electrons which were actually got excited from the photosystem 2 are now transferred to photosystem 1, which absorbs the light of 700 nanometer, and these two electrons are again transferred to its primary electron acceptor. This primary electron acceptor pair is going to transfer the pair of electron to a very short electron transport system, which involves the pyridoxine as an transport complex. This ferredoxin is going to accept these two electrons and is actually now going to pass on those two pair of electrons to NADP, right? Now these NADP is actually going to now be converted as NADPH plus H plus. That means it is also going to receive in this process the two H plus. Now where does these H plus come from and how these reduction? Again, this is also the energy source, right? Let us skip it here, stop here and move back to this chlorophyll A molecule of the first photosystem. As I told you earlier also, the non-cyclic photophosphorylation is going to start with PS2, which has a reaction center, chlorophyll A molecule absorbing 680 nanometers of light. Now, the electron that every time chlorophyll A molecule is transferring to the electron acceptor should also be replaced. Sorry, not reduced, it should be replaced. Continuous energy supply during the daytime, continuous light supply during the daytime is continuously exciting the electrons, and these electrons are being transferred to the primary electron acceptors. But then these chlorophyll A molecules should also have the electrons that should replace the electrons that are being transferred. So for that, what happens here is there will be splitting of water molecule taking place near to photosystem 2, where the water molecule is split by these photolysis. Again, in the presence of light, this reaction is going to take place. So photolysis, in the presence of light, lysis will take place. So it is a breakdown of water molecules will take place to release two electrons, protons, and the oxygen molecule will be released, right? So these oxygen is going to act actually as a byproduct which is released during the process of splitting of water molecules. So the oxygen which is released during the process of photosynthesis, when you write the reaction, CO2 plus water releases C6H12O6 plus O2. So that O2 is actually released from the water molecule. This one pair of electron that is formed here will be replacing for every molecule or every electron pair that is transferred to the primary electron acceptor. And this 2H plus, which are formed as a result of splitting of water molecules, will be actually going to this NADP, where it will reduce the NADP to form the NADPH. It can also be written as NADPH2. So both the form of storage energy are now formed as a result of the photophosphorylation reaction. Let me write here this. FD is ferrodoxin. So you can say that the chlorophyll A, photosystem 1 has its own electron transport system, as well as the photosystem 2 has its own electron transport system. Continuous supply of electrons to photosystem 1 is from the photosystem 2. So every pair of electron getting excited at the reaction center of photosystem 1 is being replaced by the pair of electrons that come from photosystem 2. And for every pair of electron that is excited from the photosystem 2 is replaced by splitting of the water molecule, and the protons that are formed here is going to reduce the NADP. Now, this is all about your non-cyclic photophosphorylation. Now, if you see here, why it is called as non-cyclic? Because electrons once excited from the photosystem 2 are nowhere returning back to the system. So, it is called as the non-cyclic photophosphorylation. And if you pay attention here, the electrons are transferred in this manner. That is the reason the non-cyclic photophosphorylation is also called as the Z scheme. Fine? The electrons, remember, the electrons always will be transferred to the ETS in a downhill manner. That means the energy will be always reduced. Here they again get excited because at the reaction center, chlorophyll A molecule is again going to absorb light of 700 nanometer and hence they get excited. Now, where exactly does this non-cyclic photophosphorylation take place? Let us see that. The process of non-cyclic photophosphorylation occurs in thylakoid of the chloroplast. Fine. Within the thylakoid, there is chlorophyll A molecule which is going to trap light, and the electrons will get excited to synthesize ATP and NADPH. So you can say that synthesis of ATP is taking place where? Within the thylakoid region, right? But the conversion of these and reduction of these NADP to form NADPH is possible because of an enzyme called as NADP reductase. These NADP reductase enzyme is actually present in the stroma. So you can say that reduction of NADP is going to take place in the stroma part. So if I draw a diagram here for the chloroplast, the double membrane structure, here there is the grana. Okay, this is the thylakoid. So within the thylakoid, the splitting of water molecule as well as the transfer of electron is taking place. So you can say that the oxygen is released within the thylakoid, whereas the photosystem 1 will be present at such location that the NADP which is formed will be reduced by NADP reductase in the stroma. Here is formation of NADPH2. So here there is the first part of the reaction taking place, and the last part or the other form of the energy source is actually formed in the stroma. Sorry, my mistake. Let us consider this to be the grana of the thylakoid. So this is the oxygen. And if this is the complete chloroplast, then this will be the stroma. This is the grana, or you can say the thylakoid, inside which the oxygen is released and the electrons get excited. Whereas the NADP is released on the surface, and outside in the stroma there is NADP reductase enzyme which is going to reduce or convert these NADP into NADPH2. So this is all about your non-cyclic photophosphorylation. It is called as non-cyclic because the electrons once released from the reaction center are never going to pass or return back to the reaction center. And remember, the non-cyclic photophosphorylation involves two photosystems, photosystem 2 and photosystem 1. It starts with photosystem 2 and the electrons then will be transferred to photosystem 1. Thank you very much.